Hello everybody! Welcome back to Catalysis, Discovery and Technology Tool, where we discuss and explore catalysis and its future in science and medicine. In this lesson, we will explore enzyme catalysis. So, let's get right into it. This lesson will answer the questions: how do enzymes work? What are the types of enzymes? How do we quantify enzymes? And how do we quantify enzyme inhibition? How do enzymes work? At a macroscopic level, enzymes work very similarly to metal catalysts. Microscopically, it is very different. Enzymes are homogeneous catalysts. The vocabulary for enzymes is a little different though. Generally, any reactants are referred to as substrates. Products are still called products. The substrate and enzyme interact to form an enzyme-substrate complex. The complex then goes on to form the product. This also releases the enzyme. Sometimes, however, the complex dissociates back to the substrate. Put more simply, an enzyme and a substrate react to form an enzyme and a product. This is the most frequent path that an enzyme can follow. The sequence of basic steps is known as the mechanism. Adding up all these basic steps must give the overall reaction enzyme to product. There are many types of these mechanisms. Sometimes the enzyme forms a complex with the product. Sometimes the product can revert to the substrate. Note that in principle, the substrate can directly convert to the product without the enzyme. This reaction will be so much slower that we can assume it does not happen. This turns out to be a very good assumption. This is a simplified but surprisingly accurate representation of what is actually going on. In reality, enzymes are proteins. These are very large complex molecules with molecular weights of about 10,000 up to 1 million. The structure of proteins is a study in and of itself. Human cells have about 1,000 different enzymes in each cell. Essentially, the protein holds the substrate in such a way that the substrate is more active, reducing the activation energy. This is the most important thing about enzymes when studying catalysis. What are the types of enzymes? Enzymes are categorized in many ways. The simplest is by looking at the type of reaction they catalyze. A and B can be basically anything. The definition is purposefully broad to make categorizing enzymes easy. Oxidoreductases. Oxidoreductases catalyze the transfer of electrons from one molecule to another. The molecule losing electrons has its charge increase. It is oxidized. The molecule gaining electrons has its charge reduced. It is reduced. Above A is oxidized. Its charge is increased by 2. B is reduced. Its charge is decreased by 2. Since A is oxidized and B is reduced, This is called a redox reaction. That is why the enzymes are called oxidoreductases. A common example of an oxidoreductase is alcohol dehydrogenase. The enzyme converts an alcohol to an aldehyde. Transferases. This is a simpler reaction. The functional group A is transferred from B to C. A functional group is a commonly appearing substituent of a molecule. Examples of functional groups are hydroxyl, fluoro, phenyl, and many more. An example of a transferase is methyltransferase. This enzyme transfers one carbon, a methyl group. Hydrolases. Hydrolases break chemical bonds using water. Here, the bond between A and B is broken. This is known as hydrolyzing the bond, hence the name hydrolase. A common example of a hydrolase is lipase. Lipase is essential to the digestion of fats, also known as lipids. A common non-biological example of a lipase is in modern enzyme-based detergents. Lipase breaks water-insoluble fats down to soluble fatty acids and glycerols that can be washed away. Isomerase. Isomerases rearrange a molecule. Isomers are two different molecules that have the same formula but different structures. An example of this would be rearrangement of N-hexane and 3-methylpentane. Both molecules have 6 carbons and 14 hydrogens, so the formula for both is C6H14. However, they are arranged differently and will behave differently both physically and chemically. Lyase. Lyases catalyze the breaking of bonds. Other than when done by hydrolysis. Hydrolysis is such an important reaction that it gets its own category of enzyme. Lyases typically break bonds in large molecules, so the examples here are a little more complicated. Glutamate decarboxylase leaves a carbon-carbon bond. Ligase. Ligases catalyze the formation of bonds, usually bonding two large molecules. They are categorized by the type of bonds that they form. Sometimes, a smaller molecule is released to form a bond between larger molecules. Sometimes, ATP is consumed to form the bond. DNA ligase is an example of a ligase. It catalyzes the combination of two strands of DNA. This is done artificially to join strands during the creation of recombinant DNA or naturally to repair damaged DNA. Translocase. This is the 7th category recently recognized August of 2018. Translocase catalyzes the movement of molecules or ions across membranes. These are not traditionally reactions, as the reactants and products are usually the same molecule. Translocases reduces the energy needed to cross the membrane. How do we quantify enzymes? The purpose of an enzyme is to catalyze a reaction. To quantify enzymes, we look at their effect on reaction rates. Mathematically, the analysis of enzymes is similar to other catalysts since the fundamental steps are similar. To review the steps, the enzyme and the substrate form a complex. The enzyme-substrate complex reacts to form the product and release the enzyme. There are other, more complicated mechanisms, but we'll start with this one. Let's look at the math behind this. Write out rate laws for each step. Sometimes it's helpful to write out reversible reactions as two reactions. Assume that the net rate of formation of any active intermediates, any complexes, is equal to 0. This is known as the pseudo-steady state hypothesis. This is the same assumption made during the analysis of heterogeneous catalysts. Here, we only have one reaction intermediate. Active intermediate is formed in reaction 1 and consumed in reaction 2 and 3. Solve for the concentration of active intermediate. We will now do an enzyme balance to eliminate all unknowable concentrations. While we can find the concentration of total enzyme and the concentration of substrate, it is not possible to quantify the different concentrations of enzyme, bound to substrate and not, as well as the reaction constants. However, we do know that the total concentration of enzyme includes that bound and not bound to the substrate. From before, we know that the concentration of enzyme-substrate complexes is equal to K1 Ce Cs over K2 plus K3. Following some rearrangement, we can therefore quantify the concentration of enzyme unbound to substrate as Ce total over 1 plus K1 Cs over K2 plus K3, a value in all knowable concentrations. Next, substitute this into the law for the production of our product. Rp is equal to R3, which is equal to K3 times the concentration of the enzyme-substrate complex after substituting the same value for CES as before and after substituting in the value for the unknown CE that we found in the last step. We can get the rate of the production of our product all in the known concentrations and reaction constants, or K1, K3, Cs, Ce total all over K2 plus K3 plus K1 times Cs. We could stop here, but this is too many unknowns that aren't concentrations, K1, K2, and K3, that we would have to linearize data to solve for. A simpler method is to combine these reaction constants into one unknown value. First, let's see what our maximum rate is. That will occur as the concentration of our substrate approaches infinity, as our enzymes will have the greatest possible substrate to react with. Vmax is equal to the limit of Rp as Cs approaches infinity, which we know to be K3 times the total concentration of enzyme. Let's group the remaining constants together and call them Km. Rp, which is equal to K1 Vmax Cs over K2 plus K3 plus K1 Cs, following substitution of Vmax in for what was previously K3 Ce total, is also equal to Vmax times Cs over K2 plus K3 over K1 plus Cs after dividing by K1. Now, all of the rate constants can be combined to form Km, thus making the rate of production of our product equal to Vmax times Cs over Km plus Cs. Now it is very, very important that we don't forget our units. Take a look at this table, for example. It will help you as we move forward. Concentration can have many units, but most notably, it is in terms of mass per unit volume or moles per unit volume. Molarity. Reaction rates are usually in terms of concentration per unit time, and reaction constant units are dependent on the reaction order, Essentially, they are whatever unit it takes to balance out the concentration units to make the overall reaction rate units correct. Now we have our simplified equation. Km is known as the Michaelis constant. This rate law is known as Michaelis-Menten kinetics. Now we have only two unknowns, Km and Vmax. We know what Vmax means physically, but what does Km mean? Looking at the rate law, Km must have the same units as Cs since they are added together. So what happens when they are equal? So when Cs is equal to Km, the rate is half of its maximum. This is easier to see on a graph. We can see from the graph that the maximum rate is approximately 5. That means that Vmax is approximately 5. We'll see how to determine this exactly in a bit. At what concentration does the rate equal half of the max, 2.5? That concentration should be Km. According to this graph, Km equals 0.5. Let's look back at what values we lumped into Vmax and Km to see what we can do to change these, if that's needed later. So if we increase K3, or if we increase the total concentration, Vmax will also increase. Importantly, increasing the amount of enzyme will increase Vmax. Km equals K2 plus K3 over Km. The only way to change rate constants is to change the temperature. This tells us that for a given enzyme at a given temperature, Km should not change. Even adding more enzyme will not change Km. Let's look at two experiments with different amounts of enzymes. One experiment has 1 gram of enzyme. The second has 2 grams of enzyme. This is the data from earlier. Vmax is approximately 5 and Km is approximately 0.5. The yellow line is the data with twice as much enzyme. We expect the max rate to double, but the Michaelis constant should be the same. Let's check to see what the concentration at half of the maximum rate is, as we did earlier. Even though the maximum rate changes, the concentration of substrate at which we reach half the maximum rate is the same. This is a little strange to think about, but referring back to the plots helped tremendously. Suppose we couldn't see what the maximum rate is. Not only do we not know Vmax, we couldn't see what Km is. How can we determine these values more quantitatively? Same as for heterogeneous catalysis, linearization. In fact, the same linearization works since the rate law is basically the same. We invert both sides of the equation. The slope will be Km over Vmax, and our y-intercept will be 1 over Vmax. The blue line, 1 gram of enzyme, has a slope of 0.1 and a y-intercept of 0.2. Vmax equals 1 over 0.2 equals 5. Km over Vmax equals 0.1 equals Km over 5, which equals Km equals 0.5. The yellow line, 2 grams of enzyme, has a slope of 0.05 and a y-intercept of 0.1. Vmax equals 1 over 0.1 equals 10. Km over Vmax equals 0.05 equals Km over 10, which equals Km equals 0.5. Plots like this are used so commonly that they are given a name. These plots are called Lineweaver-Burk plots. So we can see that even though the maximum rate increased, we do indeed get the same Michaelis constant for both experiments. Similar to heterogeneous catalysis, we propose a mechanism. We propose the simplest mechanism leading to Michaelis-Menten kinetics. There could be multiple reactants. The product could bind to the enzyme, and the reaction could be reversible. Enzymes are quantified by their Michaelis constant and maximum reaction. The maximum reaction rate is dependent on the amount of enzyme. The Michaelis constant is the concentration at which the reaction rate is half of the maximum. This does not depend on the amount of enzyme. So what else can happen? Other chemicals can interfere with this process, binding up the enzyme. This is known as enzyme inhibition. How do we quantify inhibition? Inhibition occurs when any chemical serves to reduce the activity of the enzyme. Inhibition is not always a bad thing. Many drugs are designed to slow undesired reactions by inhibiting approximate enzymes. A classic example of therapeutic enzyme inhibition is the treatment of methanol poisoning. Methanol poisoning occurs when methanol is ingested. Methanol is decomposed by the enzyme alcohol dehydrogenase to form formaldehyde and formic acid. Formic acid can cause blindness and even death. To inhibit this reaction, methanol poisoning is treated with ethanol. Alcohol dehydrogenase is found primarily in the liver and will preferentially bind to ethanol over methanol. The methanol, since it cannot bind with alcohol dehydrogenase, leaves the liver unreacted and is excreted in urine. To understand the effect of an inhibitor, we do as we did before and propose a mechanism. Let's consider the example of methanol poisoning. In this case, methanol is the substrate, ethanol is the inhibitor, and alcohol dehydrogenase is the enzyme. Since we have more species now, I will just use E to denote the concentration of enzyme instead of CE, and so on with other species. So let's see what these actually look like. There's quite a bit of algebra to lead to the rate law, but the same steps are used. Apply the pseudo-steady state hypothesis to get the concentration of active intermediates. We now have two active intermediates, enzyme and substrate, and enzyme and inhibitor. Now let's draw some comparisons. Now we do a total enzyme balance. Can you see where we simplified this? Write out the rate law for a product. Sub in the relation for enzyme from the enzyme balance. Now we have our equation. The only new definition here is Ki equals K5 over K4. This value describes the enzyme affinity for the inhibitor. High values mean the inhibitor has low affinity for the enzyme. So what is the effect on the rate? Here, the blue line is the uninhibited, and the yellow is the inhibited. The rate is reduced across all concentrations. Looking at the rate law, at high concentrations of substrate, the rate will still approach the same maximum rate. The effect is that Km is increased. It takes a higher substrate concentration to reach half the maximum rate. In this case, since the substrate and inhibitor are competing for the same enzyme, this is called competitive inhibition. There are two other types of inhibition commonly seen. Uncompetitive. The inhibitor binds with the enzyme substrate complex. And non-competitive. The inhibitor can combine with either the enzyme by itself or the enzyme substrate complex. All of these effects are more easily shown on a Lineweaver-Burk plot. First, let's look at the mechanism for each case. Second, let's look at the rate laws for each case. A step-by-step derivation will not be done for all types of inhibition. Let's take a moment and compare uncompetitive and non-competitive reactions. Uncompetitive reactions, the inhibitor binds only with the enzyme substrate complex, and in non-competitive reactions, the inhibitor can bind to either the enzyme or the enzyme substrate complex. So let's look to see how these differences can translate onto a Lineweaver-Burk plot. In a competitive reaction, the slope has increased. In an uncompetitive, the y-intercept has increased. And in non-competitive, both the slope and y-intercept have increased. Remember, on a Lineweaver-Burk plot, the y-axis is the inverse of the rate, so being higher up on such a plot means the rate is actually lower. First, let's look at the plot of rate versus concentration. Competitive. Km is effectively increased. Vmax is not changed, but the reaction takes longer to get to Vmax. Uncompetitive. Vmax is effectively decreased. The reaction never goes as fast as the inhibited reaction, but it gets to half the lower Vmax just as quickly. Mixed. Vmax is effectively decreased and Km is effectively increased. The reaction never goes as fast as the inhibited reaction, and it takes longer to get to half the lower Vmax. So what did we learn today? Enzymes are proteins that catalyze reactions. Proteins are large, complex molecules, and there are many different enzymes in each cell. There are 7 as of August of 2018, and only 7 types of enzymes. Enzymes are quantified by their Michaelis constant. This represents the concentration of substrate needed to reach half the maximum rate possible. Enzymes can be inhibited. There are 3 primary types of enzyme inhibition. Competitive effectively increases Km. Uncompetitive effectively decreases Vmax. And non-competitive, or mixed, effectively increases Km and decreases Vmax. Well, that's all we have today. Thank you so much for watching, and as always, have a good one.